Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Hello everyone. My name is Mia Rahmawati. I am from Biology Education Class C, Faculty of Mathematics and Natural Science, Semarang State University. Today, I am going to talk about effect of temperature on enzyme activity. Okay, I'm going to give presentation about effect of temperature on enzyme activity by Mia Rahmawati with lecture Miss Lutfia Nurhandianti, SPD, MPD. Okay, the presentation will be in 6 parts. First is introduction, second is background, third is method, fourth is result, fifth is discussion, and the last is conclusion. Okay, first, I'd like to talk about introduction on this practicum. Enzymes are the most abundant protein group in living cells. Enzyme synthesis occurs within cells and most enzymes can be obtained by extraction from tissue without damage. As catalysis, enzymes are different from simple inorganic and organic catalysts, which generally can catalyze various chemical reactions. Enzymes have very high specificity, both for the reaction or substrate and the reaction in catalyst. Then enzymes can increase the reaction rate tremendously without the formation of byproduct and molecules in aqueous solution under normal or physiological pressure, temperature, and pH condition. So, enzyme activity can be influenced by several factors. One of these is temperature. Second is background. This practicum is conducted for knowing the effect of temperature on enzyme activity. Can temperature speed up activity or slow it down? Okay, we will discuss this problem in the next slide. Okay, now I'd like to move on to method. Practicing the effect of temperature on the activity of this enzyme use a method that is in accordance with the practical guidebook for the enzyme chapter. In addition, this practicum also use experimental materials and tools. So, in this method, we will discuss the materials, tools, needs, and the work steps that must be done. Okay, first is ingredients. In this practicum, several materials are needed including enzyme amylase, 2% starch amylum, iodine reagent, Benedict reagent. Okay, second is tools. Well, the tools needed are test tube rack, test tube, water bath, thermometer, drop pipettes, beaker glass, and drip plates. Okay, and the next we will discuss the working steps. 1. Prepare 5 clean and dry test tubes. Label each test tube with a number. 2. Prepare the amylase enzyme or saliva obtained from gargling with clean water, then rinse again with 20 ml of 0.2% NaCl, after which it is collected. Third, in each 1, 2, 3, 4, and 5 test tubes, 2 ml of 2% starch solution was added. Then 1 ml of amylase enzymes was added to each tube. 4. Test tube 1 is put into a glass beaker filled with ice water. 5. Test tube 2 was stored at room temperature. 6. Test tube 3 is put in a water bed temperature of 37 until 40 degrees Celsius. 7. Test tube 4 is placed in a water bed temperature of 75 until 80 degrees Celsius. 8. Test tube 5 is put into a boiling water heater. And the last, each tube in place let stand for 15 minutes. Then test with iodine and Benedict reagent. Okay, the next is test with iodine and Benedict reagent. First is test iodium with work steps. First is prepare a clean and dry droplet. 2. Number the indentation of the triplet with the number first until 5. 3. Drop each 3 drops of the solution from the previous treatment according to number. 4. In each indentation of the triplet from number 1 until 5, each insert drop of reagent iodine. After that stirred. 5. The color product or appears on each droplet number 1 until 5 in the table result of observation 1.1. Okay, and the next is test Benedict with work steps. First is prepare a clean and dry test tube and label it number 1 until 5. 2. Tubes 1, 2, 3, 4, and 5 enter each 0.5 ml of the result of the effect experiment temperature on the activity of the enzyme according to the tube numbering on the result of treatment early 1. 3. In each tube drop 2.5 ml of Benedict reagent. 4. Shake the tubes 1 until 5. Then heat each tube in a water bed for 5 minutes. 5. Observe the color of the solution and the precipitate in each tube. And the last, write down the result in the table of observation 1.2. After that is result. First result, test iodium. In the iodine test, it produces a color in the tube 1 is brownish purple, tube 2 black tan, tube 3 brown, tube 4 purple black, and tube 5 is purple. And the next is result test Benedict. The Benedict test produce a color at tube 1 is blue and there is no sediments. The tube 2 color is green and no sediments. Tube 3 color is green and there is a color precipitate is brick red. And the tube 4 color is blue and the last tube 5 color is blue, and the tube 4 and tube 5 is no sediments. Then we will discuss the result of the iodium test table and Benedict. First is test iodium. The iodine test is used to detect the presence of starch in the iodine test. The method uses a previously treated solution or amylase enzyme and starch solution heated at different temperature. By adding 1 drop of iodine reagent in each test tube, which produce Test tube 1 produces a brownish purple color, which means at test tube 1 contains starch. Test tube 2 produces a brownish black color, which means test tube 2 contains starch. Test tube 3 produces a brown color, which means that test tube 3 does not contain starch. Test tube 4 produces a purple black color, which means test tube 4 does not contain starch. Test tube 5 produces a purple color, which means test tube 5 contains starch. The color result indicate that the positive solution contains starch, indicate when the color of the solution changes or not the same as the color of the iodine solution, which is yellow-brown. Okay, and the next we will discuss the result of the test Benedict. Benedict test is used to detect the presence of reducing sugars. In the Benedict test experiment using a solution of the previous treatment or amylase enzyme and starch solution had at different temperature, and Benedict reagent which was heated again for 5 minutes to produce. Test tube 1 produces a blue color and there is no precipitate, meaning that tube 1 does not contain monosaccharida or disaccharida. Test tube 2 produces a green color and there is no precipitate, meaning that tube 2 contains both monosaccharida and disaccharida. Test tube 3 produces a green color and there is a brick red precipitate, meaning that in tube 3 it contains monosaccharida or disaccharida. Test tube 4 produces a blue color and there is no precipitate, meaning that tube 4 does not contain monosaccharida or disaccharida. Test tube 5 produces a blue color and there is no precipitate, meaning that tube 5 does not contain monosaccharida or disaccharida. The color result and the presence or absence of a precipitate indicate that a solution containing monosaccharida and sugar is indicated by the color of the solution turning green and the presence of a brick red precipitate. A positive test is indicated when the solution turns green and a brick red precipitate is present. From the discussion on the previous slide, it can be concluded that the enzyme is active at the optimum temperature, 37 until 14 degrees Celsius. At low temperature, enzyme inactive, while at high temperature, the enzyme will denature. So many explanations regarding the effect of temperature on enzyme activity. I hope that I say can be useful for all of you. If there is a wrong word in the delivery, I apologize. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon. Thank you. Goodbye.